The overall goal of the following experiment is to generate RNA aptamers in vitro for HIV-1 gp120 protein using a nitrocellulose membrane. This is achieved by PCR and subsequent in vitro T7 transcription to prepare the initial DNA and random RNA library. As a second step, the mixture of the target protein and RNA library is applied onto a pre-wetted nitrocellulose filter, which allows isolation of bound RNA molecules from unbound RNA species. After 12 selection cycles, the RNA library is cloned and sequenced in order to identify the individual RNA aptamer candidates from the library. Results are obtained that identify several new aptamers with nanomolar affinity based on gel shift assays and confocal microscopy.
The main advantage of this technique over existing measures like beads or column-based isolation measures is simple and doesn't require a pre-treatment of the protein or libraries, such as the immobilization of the protein on the beads.
The RNA library for the isolation of anti-HIV gp120 RNA aptamers is prepared from a commercially synthesized DNA library that contains 50 nucleotides of random sequences. The single-stranded DNA oligo library sequence is 81 nucleotides in length, as shown in this diagram. The random region is flanked by constant regions, a 32-mer at the 5' end and a 16-mer at the 3' end. The constant regions include the T7 promoter for in vitro transcription and a 3' tag for RT-PCR. Amplify the single